Hello, everyone. Welcome to lecture number 3 in our Medical Biochemistry series on the chemistry of carbohydrates. I am Dr. Muzamil Liaqat Ali, Assistant Professor of Biochemistry. In the previous two lectures, we covered the basic definition, classification, isomerism, and cyclic structures of monosaccharides. Today, we move forward to sugar derivatives, physiologically or biochemically important pentoses and hexoses, and the clinically relevant chemical properties of the monosaccharides. These topics have direct application in understanding metabolic disorders, diagnostic lab tests, and the structural basis of the glycoproteins and glycolipids. So let's begin. Sugar derivatives are formed by modification of the carbonyl and hydroxyl groups of the monosaccharides. Let's start with the sugar alcohols or alditols. The aldehyde or ketone groups is reduced to a hydroxyl group, eliminating the carbonyl. Example, ribitol from the ribose, which is a component of flavin coenzyme, that is FMN and the FAD. The next is the sugar acids, formed by oxidation of the aldehyde at the carbon number 1 of the primary alcohol at the position number 6 of the carbon, right? So oxidation of C1 aldehyde that leads to aldonic acid, that is gluconic acid production, right? And the next is the oxidation of C6CH2OH that leads to formation of uronic acid. The example that the glucuronic acid, important in detoxification, and glycosamine glycans, right? These derivatives are crucial in conjugation reaction and extracellular matrix formation. Now, let's discuss the amino sugars. Amino sugar results when a hydroxyl group, usually at the C2, is replaced by an amino group that is NH2 group, right? The most common example is the glucosamine. The amino group is frequently acetylated to form an acetyl glucosamine, that is GLZNAC, right? A key building block in chitin. The bacterial cell wall and the glycosamine glycans, such as hyaluronic acid and chondroitin sulfate. Amino sugars play a vital role in cell surface recognition and bacterial peptidoglycan structure. Now, the sialic acid or N-acetyl neurominic acid, right? N-acetyl neurominic acid, commonly known as sialic acid, is a 9-carbon amino sugar derivative. It is frequently found as a terminal sugar in oligosaccharide chains of glycoproteins and glycolipids on the cell surfaces. Because its carboxyl group dissociate at physiological pH, sialic acid imparts a negative charge to glycoprotein. This negative charge is essential for preventing unwanted cell aggregation, influencing blood group antigens, modulating immune recognition and viral attachment. The removal of sialic acid by neuraminidase exposes underlying sugars and can trigger clearance of aged red blood cells. Now, let's discuss the pentoses of biochemical and physiological importance. Several pentoses play a critical role in human metabolism, right? The very first one is the D-ribose. D-ribose, structural component of nucleic acid, RNA and DNA, and coenzyme like ATP, NAD+, NADP+, and FAD. Ribose 5-phosphate is a key intermediate in pentose phosphate pathway. Next is the D-ribulose. D-ribulose formed during metabolism. Ribulose 5-phosphate is an important intermediate in the pentose phosphate pathway. Next is the D-arabinose, present in gum, arabic, and the glycoprotein. The D-xylulose or xylose, found in wood gums, proteoglycans, and glycoproteins. Next is L-xylulose, that is an intermediate in the uronic acid pathway. Its Excretion in urine is characteristic of essential pentosuria. That is a benign condition. The next one, or the last one, is the D-xylose, lysose. D-lysose that is present in heart muscles and lexoflavin isolated from the human heart tissue. These pentoses highlight the central role of the pentose phosphate pathway in generating NADPH and ribose for nucleotide synthesis. Now, let's discuss the hexoses of biochemical and physiological importance. The very first one is the D-glucose, the principal sugar of the body. It is absorbed from the fruit juices, starch, sucrose, and lactose hydrolysis. Glucose is the main sugar transported in the blood and used by the tissues. Elevated blood glucose levels, that is also known as hyperglycemia, can lead to glycosuria and diabetes mellitus. The next one is the D-fructose, found in fruit juices and honey, also produced by hydrolysis of sucrose and inulin. Fructose is converted to glucose in the liver and intestine, and hereditary fructose intolerance, that is aldolase B deficiency, causes fructose accumulation, hypoglycemia, and liver damage. Fructose metabolism bypasses phosphofructokinase 1 regulation, which is clinically significant in metabolic syndromes. Next is the D-galactose. D-galactose derived from the hydrolysis of lactose, the milk sugar. It is converted to glucose in the liver and used in mammary gland to synthesize lactose for milk. Galactose is a constituent of glycolipids and glycoproteins. Failure to metabolize galactose leads to galactosemia, which can cause cataracts, liver damage, and mental retardation if that condition is untreated. The next is the mannose, the D-mannose, obtained from the hydrolysis of plant mannans, and gums. It is an important component of many glycoproteins and participates in unlinked glycosylation. Mastering these hexoses is essential because their metabolic disorders like the galactosemia, fructose intolerance frequently appear in clinical conditions or clinical scenario given in the exam. Now, the biomedical importance of carbohydrates. Carbohydrates are widely distributed in plants and animals and serve both structural and metabolic roles. In plants, glucose is synthesized via photosynthesis and stored as a starch and used to form cellulose. In animals can synthesize carbohydrates for glycerol and amino acid production, right? The gluconeogenesis. But most dietary carbohydrate ultimately originates from the plants. This underscores the fundamental position of carbohydrates in the global carbon cycle and the human nutrition. Now, let's discuss the chemical properties of monosaccharides. Monosaccharides show several characteristic chemical reactions that are exploited in lab diagnosis and metabolic studies. The very first one is the Molich test, formation of furfural derivatives, the reducing and non-reducing properties as shown in the Benedict test. The osazone formation, inidiol formation, oxidation reactions, right? The formation of sugar acids, the reduction reactions like the formation of sugar alcohols, formation of amino sugars and esters, and glycoside formation. These properties form the basis of classic qualitative and quantitative tests still used in clinical biochemistry laboratories, right? Now, let's discuss reducing sugars. A sugar is a reducing sugar if it has free aromatic hydroxyl group, the free OH at the carbon number 1 in aldoses, or the C2 in case of ketosis. And that aromatic hydroxyl group that can open to form a free aldehyde or ketone. This allows the sugar to reduce oxidizing agents such as Cu plus 2, the copper, the cuprocine, in Benedict reagent. All monosaccharides and most disaccharides, except the sucrose, are reducing sugars. The diagram clearly shows that only sugars with the free carbonyl in equilibrium are reducing. Now, the oxidation of monosaccharides. Monosaccharides are readily oxidized at their carbonyl groups. In Benedict test, the D-glucose is oxidized to gluconic acid or the D-gluconic acid, confirming it is a reducing sugar. Oxidation reactions are classified as oxidation of aldehyde group, that is C1, the aldonic acid formation or the gluconic acid formation, right? The next is the oxidation of primary alcohol at the position number 6, at the carbon 6, right? That the formation of uronic acid, the formation of glucuronic acid, that is vital for the detoxification of bilirubin and certain drugs, right? So, next is the oxidation of both ends, aldaric acid, the glucaric acid formation, right? These oxidized products have important physiological and biochemical importance, and along with that, they have important diagnostic roles, right? Now, let's discuss the reduction reaction. Both aldoses and ketoses can be reduced at their carbonyl groups to form polyhydroxyl alcohol, the sugar alcohols. For example, the D-glucose that leads to formation of glucostol, right? The sorbitol formation. The D-mannose leads to D-mannitol formation. D-galactose formation that leads to D-galactitol, that is dulcitol, right? The D-fructose that leads to sorbitol plus mannitol formation. So sorbitol accumulation in the lens in diabetic patient contributes to the cataract formation via the polyoil pathway. Mannitol is used clinically as an osmotic diuretic. So we have now completed our detailed study of sugar derivatives, that is physiologically and biochemically important pentoses and hexoses and their key chemical properties along with the certain monosaccharides. These concepts directly link to the lab diagnosis or the tests used to perform in our studies. The Benedict test, the reducing sugar, metabolic disorders like the Galactosemia, essential pentasuria, and structural biology as we discuss the Sialic acid and amino sugars role, right? So in the next lecture, we will discuss disaccharides, glucosidic bonds, and the structures of the sugars, lactose, and maltose. Thank you very much for watching this lecture. And if this lecture helped clarify the topic, please like the video, share it with your classmates and juniors, and subscribe to the channel for more high-yield, exam-focused medical biochemistry lectures. So hit the notification bell so you don't miss the lecture number 4. And I am ready for your question. So drop your question or doubts in the comment section, right? I personally read and reply to them, right? So which sugar derivative or chemical property would you like me to explain in more detail? So this is Dr. Muzamil Liaqat Ali signing off. Keep revising the structures and reactions daily. Stay consistent, stay focused, and I look forward to seeing you in the next lecture. Thank you and keep learning biochemistry with passion.